Welcome back to this channel. So today we are going to discuss about section C. Now if you haven't gone through the section D video, make sure to check it out first. Let's begin, right? As I said, section C comprises of a case-based question, and this session is for 5 marks. Case-based questions. I know all of you guys are scared about this case-based question, but trust me, you don't have to worry. You know all the concepts, right? You have studied everything. You just have to read this paragraph and apply your knowledge accordingly. I know you guys are well prepared for this exam, so you are gonna ace this part, right? Okay, so let's see what is this case study or this paragraph talking about. Okay, so they are talking about some restriction enzyme. So what does a restriction enzyme do? It basically breaks the phosphodiester bond, okay, on both the DNA strands. Perfect. Such that only one end of each molecule is cut. Okay, so what is happening here? A restriction enzyme is there. What is it doing? It basically is breaking a phosphodiester bond. Okay, so what is happening? The DNA is basically cut on one end. Okay, so these end have a single stranded DNA now. Next part, they are talking about BAMH1. BAMH1. It is basically a restriction enzyme. What is it doing? Which binds to the recognition sequence. Okay, so they have given the sequence in which this restriction enzyme is working, okay, and also telling how is it cleaving. Okay, so they cleaves the sequence just after the 5 guanine on each strand. So they have given information about this restriction enzyme, which is BAMH1, is basically cleaving in this perfect sequence, in this particular sequence, after 5 guanine. That is after 5 guanine. So here they have the ability to cleave. Okay, fine, got it. Now the question here being asked is, what is this objective of this action? What is this? For what purpose is restriction enzyme used? And all of this is happening, and gene is being, this particular DNA strand has been cut. For what objective? Why are we using this? Okay, so we are using is basically because to join the gene of interest. Okay, so you have to just mention that because we want to join the gene of interest, all of this process is happening here. Okay, this is the objective of why all of this process is happening here. Okay, main objective is basically to form a new pair of gene, that is forming a gene of interest to a particular vector or any complementary DNA strands. Alright, moving on to the second part of this question. They are talking about explain how the gene of interest is introduced into the vector. You just have to mention the process. You have studied in the chapter cloning and vector, right? The chapter 11th of our NCERT. What you have studied about the different stages, how a gene of interest is being introduced into a vector. Okay, so what happens here? In the question itself only you can get slight understanding about this particular question. You have to answer like that only. So you can tell that restriction enzyme, what is it doing? It basically separates the DNA strands. Okay, fine, perfect. Then you can talk about the DNA ligase. What does it do? It basically ligate that particular gene of interest into the vector. Okay, talk about this point and you will get the marks. Okay, next part is talking about, you are given the DNA shown below. Okay, so there is a DNA sequence. We can see a DNA sequence here. So let's see what's the question is about. Okay, so they are telling, if this particular DNA was cut with BAMH1, that is, if that DNA was cut with BAMH1, how many DNA fragments would you expect? Write the sequence of those double-stranded DNA fragments with the respective polarity. So in this question they are asking if you cut that DNA sequence with BAM-H1. What was BAM-H1? It is a restriction enzyme, right? In the question itself it is given it is a restriction enzyme, so it has the ability to cut the particular DNA sequence. So it is cutting that particular DNA sequence into fragments. So the question here they are asking is how many fragments they are cutting, and also just write down those DNA fragments with their polarity. In the question here itself only they are asking to mention polarity, but even if they don't ask in the question to write the polarity, you have to make sure that you write the polarity. Always mention the polarity, okay? Do not forget. Remember what I said, label the diagram. So consider this as also a diagram and try to mention their polarity. Okay, so let's just separate or form the fragments for this particular sequence. Now, what is cutting this sequence? A restriction enzyme called BAM-H1, right? BAM-H1 is cutting this particular sequence. So in the question only they have given which sequence do they target? This particular sequence. Where do they cleave? After 5-guanine. So in the question only they have given you an information. You just have to use this information and give the answer for this particular question. Alright, so first make sure do not get confused. You have to see for this particular sequence in this DNA strand. So let's see where this sequence is. Okay, you can see here. Okay, we can see that sequence here, and then for this part you can see the sequence here, right? Okay, now first step is to basically locate this sequence. Okay, fine, done. We have done that step. We have located that sequence. Now next step they are telling that it cleaves after 5-guanine. That is, it is cleaving after 5-guanine. Here it will cleave. Let's see. So the polarity here is 5', the polarity here is 3'. So moving from 5' to 3', you can see here after 5-guanine. So this fragment will cleave here, forming 2 fragments. Okay. Then when we are talking about down, from 5 polarity to 3 polarity, remember it's a complementary strand. Okay, so 5 polarity to 3 polarity. Okay, so after 5-guanine here, now 2 fragments is being formed here. That is your first part of the answer. That is 2 fragments. Okay, 2 fragments are formed here. Next part is asking to write those fragments. So here 5 polarity is here. Try to mention 3', 3' to 5', okay, 3' to 5', then 5' to 3'. Okay, so always mention the polarity. Do not forget about mentioning the polarity. So these 2 fragments are formed. You have to write about them. Separately, because they have asked that in the particular question using this sequence, right? Okay, moving on to the next part of this question. It was a very easy question, right? I know, like, most of the students usually gets intimidated with this 5 marks question, but it's the easiest one, I would say, because you know the concept. You have studied each and every concept, so you just have to put up your concept into this particular question. Okay, moving on, the 4th part. So here they are giving a gene M. Okay, so there is a gene M. What is it doing? It is basically introduced into an E. coli cloning vector. Okay, fine, at the BAMH1 site. So there is a particular gene M which is being introduced into this cloning vector, right? Okay, so gene M is being introduced here into this cloning vector. Okay, the question here asked is what will be its impact on the recombinant plasmid? What is recombinant plasmid? Where a gene of interest is being introduced. Okay, so here the gene M is being introduced, so this is a recombinant plasmid. Recombinant plasmid, right? Okay, so this is a recombinant plasmid. Here a gene M is being introduced. So the question asked is, what is the impact? What will be its impact if this recombinant plasmid is, that is consisting of gene M, is present? Okay, so as you know, when the BAM H1 site is being taken out and a gene M is being introduced, what will happen? The tetracycline resistant gene, tetracycline resistant gene is being cut here, right? Okay, so the tetracycline resistant gene has been cut in, okay, here, and gene M is being introduced. Okay, perfect, we understood all this. So what happens when this tetracycline resistant gene is being taken out or cut from the BAM H1 site? Basically, it will not anymore, this vector will, that is this particular plasmid will not anymore be resistant towards the tetracycline, right? So you just have to write about, there will be a tetracycline inactivation happening here as the tetracycline resistant gene is being removed and gene M is being introduced here. Okay, perfect, you just have to write about the inactivation of the tetracycline and you will be sorted for the first part. Okay, now the second part here is talking about, give a possible way by which you could differentiate a non-recombinant to a recombinant plasmid. Okay, so we have talked about the recombinant plasmid. Let's just draw about the non-recombinant plasmid. Okay, so this is a non-recombinant plasmid here. So in non-recombinant plasmid, no gene of interest has been introduced. It is exactly like that. So there BAM H1 is present and also tetracycline resistant gene is still there. Okay, so in non-recombinant, tetracycline resistant gene is still present, whereas in the recombinant plasmid, the tetracycline resistant gene is being removed as the gene M is introduced here. So as soon as both of them are being plated into a tetracycline medium, okay, both of them are placed into the tetracycline medium. Okay, so tetracycline, what is tetracycline? Tetracycline, first of all, it is an antibiotic, right? So tetracycline is an antibiotic. So this non-recombinant and the recombinant one is being placed into a medium containing tetracycline. Okay, so what will happen in the non-recombinant one, as tetracycline this is present, so in this non-recombinant they already have tetracycline resistant gene. So what will happen? They will survive, okay, they will live. And what will happen in the recombinant one? Recombinant one is not resistant to tetracycline. Why? Because the tetracycline resistant gene is being taken out. So what will happen to the recombinant one? It will eventually die. Okay, it will not produce again, it will not grow into the medium, whereas the non-recombinant will grow because they were resistant to tetracycline, because they had tetracycline resistance gene. Okay, so that is how you differentiate between the non-recombinant and the recombinant one. Okay, perfect. Let's move on to the answer and see the answer for each and every part. Okay, so the first part, they are talking about the whole objective of this particular action was to join the D gene of interest to the vector, and it will only join when the sticky end is present. You have studied about the sticky ends, right? Okay, then The second part, they are talking about the gene of interest is introduced into the vector using the restriction enzyme. What does this restriction enzyme do? It basically cuts the DNA of vector, and with the help of a DNA ligase, the gene of interest is basically ligated into the DNA of the vector. Example of restriction enzyme, again BAM-H1. If you give an example, it will be a plus point, okay? Even if it's mentioned in the question, just write it down as an example, okay? Fine. Moving on to this part C of the same question. So they were talking about the BAM-H1 will cut the particular DNA sequence into 2 fragments. You can see the fragments. Do not forget to mention the polarity. I'm again and again telling you guys, do not forget to write the polarity, okay? Okay, then the third part, they're talking about when we ligate a foreign DNA gene M at the BAM-H1 site of tetracycline resistant gene in the vector, the recombinant plasmid will lose the tetracycline resistant. Why? Because this foreign DNA is being introduced here, okay, fine. Inactivation will happen of tetracycline, and then how can we differentiate? By plating the recombinant and the non-recombinant one into the medium containing tetracycline. Tetracycline is an antibiotic where the recombinants will not grow in the medium because the tetracycline resistant gene is cut here, and the non-recombinant one will grow on the medium. Why? Because tetracycline resistant gene is present here, and then it will grow into that particular medium, okay? Okay, so this 13 question itself has an OR part, okay? So let's just try to read this case study and try to understand what is it asking us, okay? So in this GM crop, that is genetically modified crop, especially the BT crop, are known to have higher resistance, okay? So we know that BT crop is resistant, okay? So BT crop has higher resistance to any sort of pest attacks, okay? Next, they are saying to substantiate this, an experimental study was conducted in 4 different farmlands. So an experimental study was conducted in this 4 different farmlands, okay, which is growing BT and non-BT crops. Now the next part is telling the farmlands had the same dimensions, fertility, and were under similar conditions. Next is they are telling the histogram below shows the usage of pesticides on BT crops and non-BT crops in this farmland. So they have given a graph here, okay? So in this graph they are basically depicting the amount of pesticides which is used in each farmland for the BT and the non-BT crop. So you can see the BT crop is the blank one, that is this one, and the non-BT crop is the one which is having a shade. Okay, so let's just see what they are asking in the question. So in the question they are asking which of the above 4 land has successfully applied the concept of biotechnology to show the better management practice of use of agrochemicals. Okay, agrochemicals is nothing but pesticides. So as we have studied that BT crop is already resistant to the pest attack, right? So it has a certain gene, that is a resistant gene, which basically gives it resistance towards the pest. So in the question they are asking which of the 4 farmlands is having a better management, that is the better biotechnology management, okay? So here you can see that is the farmland 2 is having the better management, and let me tell you why. It's basically, you can see the BT crop is here mentioned is low, right? So what does it mean? Basically the BT crop is already resistant to the gene, so it doesn't require that much of pesticides, right? And that is why it will require only less amount of pesticides. So wherein for non-BT you need pesticides, right? Because they don't have any resistant gene present in it. That is why here you can see the non-BT is much higher than that of a BT. So farmland 2 is the one which is using the biotechnology management practice, that is the better biotechnology management practice. Okay, now let's see the next part of this particular question. So then they are asking, if you had to cultivate, which crop would you prefer, BT or non-BT, and why? Okay, so if I had to cultivate, okay, so I will definitely choose the BT crop. Why? Because BT crop, they are using less amount of pesticide, right? So of course I will use BT crop because the cost will be less. Okay, so you can answer with that. Next part is, cotton bollworms were introduced in another experiment study on the above farmlands wherein no pesticide were used. Okay, so a condition is provided where a cotton bollworm is nothing but an insect. So this insect was introduced in this particular experiment into a farmland where they haven't used any sort of pesticides. Okay, then they are asking, explain what effect would a BT and non-BT crop have on that pest, that is on the cotton bollworm. What effect will this BT and non-BT crop will have? Okay, so BT crop is already having a cry gene, right? So BT crop is already having a resistant gene, which is a cry gene. So this cry gene is basically producing a toxin, that is, it is a toxin protein. Okay, so it is having a toxin protein. So what does it do? It basically enters into the gut, and as soon as this toxin protein will enter into the gut of the cotton bollworm, it will become active. And as the gut of the cotton bollworm is alkaline, what will happen? It will basically punch hole in the gut of cotton bollworm, and that cotton bollworm will die. So that will be the effect of BT, wherein when we are talking about non-BT crop will have no effect on cotton bollworm. Why? Because non-BT crop is not having any sort of resistant gene, nor it is producing any toxin proteins. So there will be no effect on the cotton bollworm, and it will be easily be attacked by that particular cotton bollworm. Okay, so that is the answer. So you have to write it in the point. So let's see the answer. Okay, for the part A, according to the graph, farmland 2 has applied the concept because there they were using less pesticide for BT crop and more for non-BT crop, as they should. Okay, second part, if they had to cultivate BT crop, I would personally prefer BT crop. Why? Because the usage of pesticide is minimal for BT crop. And talking about the B part, that is the effects of cotton bollworm on both of them, that is BT and non-BT, follows. For in BT cotton has a cry gene. When they are introduced into them, that is BT crop is introduced into the BT crop by bacterium Bacillus thuringiensis, which causes the synthesis of a toxic protein. This protein will activate in the gut of the cotton bollworm, feeding on the cotton, thus punching holes on the lining, causing the death of the insect eventually. Okay, second part, you can mention about what effect is it having when they are eating the non-BT crop. So non-BT crop, here there is no cry gene, or there is no any sort of resistant gene which is present here. So what will happen? Cotton bollworm will eat it, and eventually non-BT crop will be succumb to the pest attacks. All right, so that's how this is the entire paper, you guys. We have finished our section C also. So in the section C, you have seen the most of the questions were from cloning vectors, and also from biotechnology. This part was from biotechnology, and there was an basic introduction about the crops also. So part C is the easiest one, you can see, because all of the students are mostly intimidated by this section. But do not get scared, you just have to read the paragraph and apply the concept which you have already studied, right? So here we have finished our entire paper. Thank you for watching this video. Now, if you have any suggestions or questions from me, make sure to put that down in the comment section, and I'll see you in the next video. Peace out.